Translation. Translation. The process of arrangement of amino acids in a specific sequence of the polypeptide chain based on the information in the mRNA is called translation. Translation takes place in the cytoplasm at the sites of protein synthesis, that is, ribosomes. Translation occurs in 4 steps. 1. Activation of amino acids. 2. Initiation of protein synthesis. 3. Polypeptide chain elongation. 4. Termination of polypeptide chain and release. Activation of amino acids. A cytoplasm consists of an amino acid pole that comprises of 20 different types of amino acids. Individual amino acids are activated by ATP in the presence of amino-acyl synthetase to form amino-acyl adenylate enzyme complex and pyrophosphate. Amino-acyl synthetase enzymes are specific for 20 different amino acids. Amino-acyl tRNA is formed by the combination of a specific tRNA molecule and amino-acyl adenylate complex. They move towards the ribosomes. Initiation of protein synthesis. Initiation of protein synthesis is introduced by the mRNA, coding for amino acid methionine. 4' group attached to methionine to form 4-methionine. 4-methionine tRNA brings the 4-methionine, which is accepted by 30S mRNA, at AUG codon near the P site. The initiator amino-acyl tRNA combines with ribosomal subunits mediated by mRNA to form initiation complex. The required energy for this process is obtained by GTP and initiation factors IF1, IF2, IF3. Smaller subunit of IF3 combines with IF3. 30S plus IF3 combines with IF1, which combines with IF2 plus GTP, which is 30S plus IF3 plus IF1 plus IF2 plus GTP. mRNA first attaches to the complex and then to the initiator. This whole complex combines with a larger subunit and forms initiation complex. It consists of F-methionine tRNA at its P site and has a free A site. GTP gets hydrolyzed to GDP and PI, releasing initiation factors IF1, IF2, and IF3. Polypeptide chain elongation. The ribosome moves along the mRNA 1 codon at a time. From the initiation codon, adding amino acids occur with the establishment of peptide bonds in the direction 5 to 3 till the termination codon. A peptide bond is created between 4-methionine at P site and the second amino acid AA2 at A site, releasing 1 water molecule. Here, peptidyl transferase acts as a catalyst. The elongation of polypeptide chain occurs in the direction C to N, that is, carboxyl to amino end. The process of bond extension continues until A site is occupied by one of the termination codons. Termination of polypeptide chain and release. Termination codons may be UAA or UAG or UGA that is present in mRNA. They do not code for any amino acid. Hence, these codons act as stop signals. RF1 and RF2 released by RF3 recognize termination codons. The polypeptide chain undergoes certain modifications. Deformylase removes the formyl group of methionine. Exopeptidase enzyme removes some amino acids from either of the ends. This linear sequence of amino acids, as shown, is known as the primary structure of the protein. This protein undergoes modifications forming secondary and tertiary structures.